SDS-PAGE stands for sodium dodecyl sulfate polyacrylamide gel electrophoresis. SDS-PAGE is a technique used to separate proteins by molecular weight. The separation is due to differential rates of migration through a polyacrylamide gel under the influence of an applied electrical field. This technique can be used to estimate molecular mass, purity, or concentration of a protein via staining or blotting. To cast a gel, you'll need gel plates, a well comb, a gel cassette, and a gel casting apparatus. First, assemble the gel plates and place them in the gel cassette. Next, place the gel cassette in the casting apparatus, ensuring a seal forms at the bottom. The gel consists of the following components: acrylamide, tris HCl, pHs 6.8 and 8.8, sodium dodecyl sulfate, or SDS, deionized water, TEMED, and ammonium persulfate. To prepare your resolving gel, combine the following reagents: H2O, acrylamide, tris pH 8.8, SDS, APS, and TEMED. Mix these by inverting the tube. Transfer the gel mixture into the gel casting cassette. Next, add a layer of isopropanol to the top of the gel solution to eliminate air bubbles. The polyacrylamide gel is a matrix formed from monomers of acrylamide and bisacrylamide. It is chemically inert, so it won't interact with proteins as they pass through. Ammonium persulfate acts as a source of sulfate radicals, which are stabilized by TEMED. Together, these are initiators of polymerization. The sulfate radical will transfer its unpaired electron to an acrylamide monomer, generating an acrylamide radical. The acrylamide radical will do the same to other acrylamide monomers, propagating chain growth. Through this process, long chains of acrylamide are generated. These long chains are cross-linked by bisacrylamide. As the polymerization process continues, a gel will be formed. When the gel has solidified, decant the isopropanol from the cassette. Stacking gel can be prepared from the following reagents: H2O, acrylamide, tris pH 6.8, SDS, APS, and TEMED. As with the resolving gel, invert to mix and transfer to the gel casting cassette. Insert the well comb into the top of the cassette and allow for the stacking gel to polymerize. After polymerization, the gel can be removed from the cassette. At this point, it's ready for use or to be stored at 4 degrees. To do this, wrap in a damp paper towel and plastic wrap. Each protein sample will have a unique amino acid sequence, and these sequences give each protein a unique shape and net charge. A protein's native structure is stabilized by covalent and non-covalent interactions between amino acids. While this structure is critical for biological function of a protein, these features will impact how a protein travels through the gel. A larger molecular weight protein, when folded, will travel through the gel like a lower molecular weight species due to its compact conformation. Correct for this effect, loading buffer will contain a reducing agent, SDS, and a loading dye. The reducing agent breaks covalent bonds that can form between cysteine residues, while the SDS disrupts non-covalent interactions such as hydrogen bonds and hydrophobic interactions. As hydrophobic residues tend to be tightly clustered, samples are boiled to aid denaturing. Protein samples will have different native charges, which will affect how they pass through an electrical field. In sample buffer, the linearized protein is evenly coated in SDS. The negative charge on SDS molecules masks the net charge of each protein. With the same mass-to-charge ratio, protein will travel through the gel based on mass alone. After samples are boiled and spun, they are ready to be loaded into a gel. To do this, secure your gel in a running module, making sure a seal is formed, and place the running module into the chamber. SDS run buffer consists of glycine, SDS, and tris HCl. The running module should be filled with this buffer as well as the chamber. At this point, the comb can be removed from the gel. Protein samples can now be loaded into the gel, leaving the first lane for a protein standard. These consist of proteins of known molecular weight to compare to experimental samples. After loading, the lid can be placed on the chamber and the electrodes can be connected to the power supply. Red to red, black to black. The run is ready to start. The tris glycine running buffer is now a gold standard for protein separation by SDS-PAGE. A key feature of this buffer system is the ability of glycine to adopt different charge states based on pH. At a neutral pH, the Zwitter-ionic form of glycine with no net charge is favored. At a low pH, the cationic form of glycine is favored, and conversely, at high pH, the anionic form is favored. These charge states affect how glycine travels through the gel. At a neutral pH, the uncharged form of glycine travels slowly through the gel, and at a high pH, the negatively charged form of glycine travels rapidly. In the two-gel system, the stacking gel has a pH of 6.8, the resolving gel has a pH of 8.8. Proteins are loaded into the neutral pH stacking gel, where the Zwitter-ionic form of glycine is favored. In the stacking gel, the protein samples will be sandwiched between slow-moving glycine and fast-moving chloride ions contributed by the tris HCl. This concentrates the samples to a uniform front as it travels through the stacking gel. When glycine reaches the higher pH resolving gel, the anion form is favored, which rapidly runs off the gel. The protein samples then migrate through the gel based on molecular weight. This process can be visualized here in a real gel. When the dye front runs off, the run can be stopped and the gel can be analyzed. Unplug the electrodes and remove the lid from the electrophoresis chamber. Decant the buffer from the running module and remove your gel. Using a prying tool, carefully separate the glass plates of your gel cassette, being careful not to tear the gel. Remove the gel from the glass plate and transfer to a container half filled with distilled water. Microwave the container for 1 minute, then place on a shaker for 10 minutes. And pour on the desired stain. Allow the gel to shake and stain. Replace stain with destain and continue shaking until bands are visible. Adding a Kim wipe to the container will speed up this process.